Hello, today I will be talking about the paper entitled Prenatal Stress Downregulates Reelin Expression by Methylation of its Promoter and Induces Adult Behavioral Impairments in Rats. So, previous studies discovered that prenatal stress causes a predisposition to emotional and cognitive impairments, which increases the risk of developing certain neuropsychic disorders such as schizophrenia, depression, and bipolar disorder. Reelin is an ECM protein secreted by Cajal-Retzis or CR cells in the brain and is involved in cortical neural connections in prenatal development and synaptic plasticity in postnatal stages. The hypothesis for this experiment was that Reelin has been shown to play a critical role in synaptic maturation and cortical elimination, and prenatal stress is thought to result in the methylation of the reelin promoter, resulting in its downregulation, which has been associated with maladaptive behaviors. So, prenatal stress in the last 10 days of pregnancy in rats will result in downregulated reelin and cognitive and behavioral impairments. Rat gestation is typically 20 days in length, and cortical development occurs during the last half of pregnancy, which is how the researchers decided on the time frame of the last 10 days of pregnancy. So, in order to perform this experiment, researchers divided pregnant rats with no apparent differences in reproduction activity into two groups. The first group is the prenatal stress group or PNS group, and the control group. The PNS group was placed in cages that would completely restrain movement without obstructing their ability to breathe for 2 hours a day during days 11 through 20 of the pregnancy as a way to induce stress. And the control group was handled for a few seconds, but then returned to their home cages and were not subjected to stress. On the 20th day of gestation, four randomly chosen dams or pregnant mothers from both the control group and four from the PNS group were euthanized in order to weigh their adrenal glands and collect histological and biochemical analysis of the fetal rat brains. The remaining dams were maintained until they could give birth. At 60 days old, the offspring were put through learning and memory tasks, and their spontaneous motor activity was observed to better understand the effects of prenatal stress on behavior and cognition. Throughout gestation, a daily weight measurement was taken of the pregnant mother rats, and in order to confirm that the pregnant rats were in fact subjected to stress, the researchers used the body weight and adrenal gland weight data to confirm this. So the data that I'm referring to can be found in figure 2, which is at the top of the middle column of the poster. It was found that the PNS or stress group had lower body weights, and their adrenal glands actually weighed 25% more than that of the control group. So, to explain this, when the body is stressed, the adrenal gland responds by producing cortisol. But if the body is constantly stressed, there's a higher demand for the gland, and that causes it to increase in size to be able to accommodate the workload. Short-term stress also suppresses appetite, and this accounts for the decrease in the PNS body weight data. So these findings combined are evidence that the PNS group was in fact subjected to stress in this experiment. Throughout or through quantitative analysis of the fetal rat brains, it confirmed that there was a significant decrease in reelin staining at CR neurons in the PNS rat group in comparison to the control group in all sections. To see if these results were paralleled by alterations to the Reelin dependent signaling pathway as well. A Western blot analysis was performed with Reelin and Reelin dependent signaling components using protein extracts from the cortex. They found that the results did in fact parallel, and there was a decrease in both Reelin and there was a decrease in the Reelin dependent signaling pathway in the stress or PNS group. This data can be found in figure 5. So in figure 5, Reelin CDK5 and P35 expression in the control is much greater than that of the stress group. Protein kinase CDK5 and its activator P35 are components of the Reelin dependent signaling pathway, confirming that the pathway was indeed altered by stress. MDAB1 protein expression is increased, indicating that neurons failed to adapt their expression to the lack of Reelin signal input, further confirming that a decrease in Reelin. The in vitro experiment showed similar changes as in the in vivo experiment. Figure 5C highlights CDK5 levels with decreased kinase activity, indicating decreased Reelin signaling. The next step of the experiment was to determine if epigenetic modifications triggered the downregulation of Reelin expression through alterations to the promoter's CpG methylation status. So a DNA methylation analysis was performed by adding primers against the Reelin promoter region, the use of PCR amplification, and use of specific restriction enzymes. Results were focused around the Reelin promoter distal to the transcription start site, and this information can be found in figure 6, which is located in the top right column of the poster. In figure 6, there was a significant difference between the PNS and control group, indicating that restraint-induced stress did increase CpG methylation. So this suggests that the downregulation of Reelin expression in PNS neurons is strongly associated with increased CpG methylation at the promoter of this gene. Moving on to the next step of determining if there was behavioral effects from the Reelin downregulation, various observational and memory tests were performed on the offspring at 60 days old. This was done by observing the spontaneous motor activity of the two groups, in which they found that the PNS group exhibited greater locomotor and rearing activity, indicating higher levels of anxiety. A passive avoidance experiment and a Morris test were also performed to analyze emotional and visuospatial memory of the rats. Both groups had identical Morris test results with similar decreases in latency, indicating that both groups evolved the same over the course of the learning period. Figure 8, which can be found in the center column at the very bottom, shows the results of the passive avoidance experiment. The offspring of the PNS mothers were unable to retain a condition response past day 4 or 5. The loss of condition response in the PNS offspring suggests a deficit in memory consolidation. To summarize the results of this experiment, Reelin expressing neuron density in embryonic day 20 brains decreased with prenatal restraint stress. Changes in the Reelin signaling pathway occurred both in vitro and in vivo, as indicated by figure 5. In addition, increased methylation of the Reelin gene promoter in the cortex of the PNS rats was confirmed through DNA methylation analysis, as stated in Figure 6, and the offspring of the rats that were subjected to prenatal stress did display increased anxiety and experienced greater emotional memory deficits than that of the control group. All of the information and data collected in this experiment does support the stated hypothesis. Um, Again, reinstating that the prenatal stress is thought to result in the methylation of the Reelin promoter, which results in the downregulation of Reelin, which causes cognitive and behavioral impairments. This experiment was able to establish a relationship between stress, prenatal stress, and the disruption of the Reelin signaling pathway through the decreased density of CR neurons. However, the decreased density of CR Reelin positive cells could also be a result of not only the downregulation of Reelin gene expression, but also through increased neuron death. While this study does support that the decreased neural density is caused by decreased Reelin expression, an additional experiment utilizing an apoptosis assay could be helpful in ruling out neuronal death as the cause of decreased Reelin expression, since prenatal stress was found to decrease intrinsic apoptosis activation. But this experiment does provide a great starting point for further understanding memory and behavioral issues that could result from prenatal stress. Thank you for watching.